Okay, thank you. Well, why don't we now start? Make sure everyone else has muted at their sites, and I'll welcome you to the DNA repair interest group. And we moved from one room to the other. We were having some technical problems before, and the talk today is Dr. John Eisen. And John, if you could, let's see if I can work this. If you could stand, let's see. You can stand.
Please, everybody mute aside from NIH Bethesda. Everybody else mute, please. Currently, I see somebody else.
There's John. He's waving. Okay. He's getting mic'd up right now. And John has made a big circle. He started here in the Bethesda area and went to the Whitman High School, and his parents actually worked here at a...
Speak up, Ken, a little louder, Ken.
And then he went to Harvard, undergraduate, working with the endo and symbiotics, and then went to Stanford for his PhD degree, and was working with Phyl Homologs on the evolution of DNA repair. In 1998, he came to the Tiger Genomics up the road a little bit, and now he's a full investigator. And his talk today is more questions than answers, insight into DNA repair processes from genome sequencing projects. So, take it away, John. Turn the lights down.
I don't know if the mic is actually working. Can you hear me?
We can barely hear you, John. You got to find a way to speak up a little more.
All right, the mic doesn't appear to be working, so. Yeah.
Right there, that was better.
How about now, Will?
Slightly better, but we'll go on.
All right, we've switched rooms and the mic does not appear. I'm not sure it's working.
Have to hold it close.
How about that?
No change.
All right.
Just speak up, John. That's okay.
I will. I'll speak up and drive everyone here crazy. How's that?
What else is new?
Yeah. All right. So thank you, Ken and Will and the other people organizing this DNA repair symposium, and apologies to everyone here who's going to run screaming from my loud voice. What I'm going to do today is something very different from what I normally do. I'm going to try and tell you a lot of the things we don't understand. I've been at Tiger for 6 years now. One of the things that I've been doing there is a lot of analysis of putative DNA repair genes in genomes. And we think we've learned a lot about DNA repair, but I'm going to try and present you with some of the questions that have come up that we haven't really been able to resolve through our analysis, or interesting things that I think are worth pursuing by someone out there in the DNA repair community. Is that any better, Will?
Marginally, yeah. Well, just don't strain your voice. We don't want to have that happen. So just we'll try. We can hear you, John. It's okay.
Okay. So what I'm going to do is give a tiny bit of background, and then go through a few different cases where we have questions that we haven't answered, or a few that we may have answered, but interesting things that have come up in analysis of genomes. And then I'm going to talk very briefly at the end about avenues to trying to answer some of these questions. So it was 10 years ago that the first complete genome sequence was determined of Haemophilus influenzae. And I was a graduate student in Phyl Homologs' lab at the time, trying to clone DNA repair genes from various organisms using degenerate PCR, and then realized that these places were going to sequence all of the genomes that I was painstakingly trying to clone the genes from. So I decided, rather than pursue that any further to try and join the machine, so to speak, and work on genome analysis at a genome center. And this is actually a slide that's a couple of years old, but it shows the progress in genome sequencing from 1995 through to 2003 just for prokaryotes. And there's been an amazing increase in the number of complete genome sequences that have been determined for prokaryotes, and it's still almost on an exponential increase in 2005. I put this slide in here to remind myself to make a point. This was a really funny cartoon that came out when the human genome was announced as being complete for the second time they announced it being complete. And the cartoonist, this says 1 human being, some assembly required. And in this picture of the human genome, there are gaps in the assembly. And that's important because actually the human genome is not even yet complete, despite, I think, the 4th announcement that they've completed the human genome. And there are many reasons why, when we analyze genomes, it's useful to have an actual complete sequence of some organism. And I could go through a litany of 20 or 30 different reasons why we like to have complete sequences of organisms. One of the ones that I'm most interested in is that it allows us to look for the absence of genes in a genome. When you don't have the complete sequence, you always wonder if the gene that you think is not there is in the missing data that you haven't yet finished. So when we sequence a genome, or when anybody sequences a genome, there are a large number of steps that people go through to try and do a preliminary analysis of the genome. You try and identify where the genes are, you try and characterize the features of the genes, you try and characterize various features of the genome. One of the things that I'm heavily involved in is trying to predict the functions of genes by various bioinformatic analyses, and then trying to use the predictions of all of... Hang on a sec. Trying to use predictions of all of the genes to predict pathways in the organism, and then a variety of other things you do once you've gotten to that point. And one of them is that we can now do much better and better is to compare genomes to each other, because there's so many genome sequences out there. And this is where I believe evolutionary analysis is most useful, because rather than just looking at the similarities and differences among genomes, trying to understand how those differences came into being through evolutionary analysis is very useful. So what I'm going to do now is first give you a case study, an old one, but I think still a very useful one to think about in terms of analyzing genome sequences and their putative DNA repair capabilities. And this came up when Tiger published the complete genome of Helicobacter pylori, the organism that is responsible for many ulcers and possibly some gastric cancers. This is a lovely picture of the beautiful organism. And so when the genome was published by Tiger, they had this paragraph in there that I found very interesting, which was that they predicted that the organism was able to perform mismatch repair because of the presence of homologs of MutS and UVRD. They noted, but weren't sure what it meant, that there were not homologs of MutL and MutH in the genome. And so what does this mean? I, as a DNA repair person trying to clone MutS and MutL homologs from lots of different organisms, all the experimental studies available at the time said that if an organism has mismatch repair, it has to have homologs of MutS and MutL. This was known in bacillus, in E. coli, in yeast, in humans, in a huge diversity of organisms. So now what do? Here's the first example of an organism that has a MutS homolog and no MutL homolog. Well, to make a long story very short, it turns out what's going on in prokaryotes is that there are 2 subfamilies of MutS homologs. Prior to genome sequencing, we only knew of one of those subfamilies, which I've called MutS1. And MutS1 includes the genes that had previously been known to be involved in mismatch repair in prokaryotes. And now there's this new subfamily, which we get more and more representatives of when we sequence genomes. And I've called that MutS2. And at the time of the H. pylori genome, there was not a single experiment known for any of the genes in this MutS2 subfamily. The the H. pylori gene turns out to be in this MutS2 subfamily. So H. pylori does not have a MutS1. It only has this weird MutS2 for which no one had ever shown the function of. So given that there are these two MutS subfamilies and that the subfamily that H. pylori gene is in has no known function associated with it, I would not predict that this organism had mismatch repair. In particular, the absence of MutL also leads me to suggest that I would not predict the presence of mismatch repair. That doesn't mean the organism doesn't have it, but the prediction can be enhanced by looking at this subfamily structure and mapping functions onto the subfamilies. Well, it turns out it looks like this was one case where we got the prediction right. Experimental studies since then have shown that H. pylori has a very, very high mutation rate in normal cells. If you knock out the MutS2 homolog from the H. pylori genome, it has no detectable effect on mutation rates, so it does not look like that gene is involved in mismatch repair. And so this was my sort of suggestion to myself that some type of evolutionary analysis can help make predictions of the DNA repair capabilities of organisms. And I outlined this as a method called phylogenomics, using phylogeny to predict the functions of genes that came out of genome projects. That's not particularly important here, but this phylogenomic approach, integrating evolutionary analysis and genome sequences, is what most of the examples I'll be giving you for the rest of the talk rely on. So now I want to go into some of the examples where I think we don't quite know what's going on, and we don't necessarily have the experiments to back it up. So here I'm going to talk briefly about what I call long-lost cousins. These are new subfamilies, like the MutS2 example, where we don't really understand what the genes in the new subfamily are doing. And I'm just going to give a couple of examples of this. So one of them is UvrA. So again, prior to genome sequencing, we only knew of genes in the UvrA1 subfamily, or we only had experimental studies, certainly of genes in this subfamily. As more and more genome sequences have come out, we found that there's a second subfamily of genes related to UvrA. Some species have both UvrA1, which is the normal nucleotide excision repair UvrA, and some genomes have a second UvrA homolog, which I've called UvrA2. There have been a few studies now of the UVR2 and Deinococcus radiodurans, but we found UvrA2s in about 15 genomes now, and we really don't understand what this subfamily of UVRA relatives is doing, whether they're involved in some alternative excision repair pathway, or whether they're involved in recruiting other genes to DNA damage spots. It's not entirely clear. One suggestion we made, which there's still no experimental evidence at all related to, but I'm going to just put it out there for people to think about, is that people may not be aware that the UVRA genes are actually related to ABC transporter proteins. They've got some core motifs that are shared with ABC transporter proteins. And so we proposed that possibly maybe the UvrA2 subfamily is involved in somehow exporting DNA damage products out of the cell. So linking nucleotide excision repair to some sort of transport pathway to get rid of things that might potentially damage the cell again. So here's one where we have some experimental studies of one gene in the new subfamily, but not many of the other ones. So there was a paper a few years ago by Molinar et al. Showing that there's an alternative excision repair pathway in E. coli, and that one of the genes involved in this they called CHO. CHO is actually distantly related to UVRC. It shares a motif/domain with UVRC. And when you do an evolutionary tree of this CHO UVRC family, you find again that there are a large number of genes that appear to be more closely related to CHO than they are to UVRC. And there's very little experimental information known for the genes in this CHO subfamily, except for the E. coli member of the subfamily. So again, do all of these other genes in this subfamily have a similar function to the E. coli CHO? It's not entirely clear, and genome analysis suggests that these are worth pursuing for some type of experimental study. I have 1,000 examples like this. I'm not going to belabor the point with every single example we can come up with. There's certainly another one in a lot of genomes that we've looked at in terms of photolyzed homologs. This is just an example from Vibrio cholera, which actually has 3 photolyzed homologs in the genome. It's not clear what the function of all of these are. Some of them appear to be more closely related to 6-4 photolysis. Some of them appear to be more closely related to CPD photolysis. These are all in the class 1 photolyzed family, I believe. But again, when we start to explore genome space, we discover lots of diversity out there, both in terms of extra subfamilies within a previously characterized family or superfamily of proteins, as well as multiple genes within one genome that we don't know whether or not they have different functions biochemically, or if they're targeted to different parts of the cell, or exactly what's going on. Another important thing that has been discovered from genome analysis is that there's lots of DNA repair genes that have novel domain sharing patterns relative to what was known prior to the genome sequencing projects. This is just one example that I found from a long time ago. The ADA gene, which is involved in alkylation repair in E. coli, has these 2 motifs, the transcription regulatory motif and the methyltransferase motif. OGT genes have just the methyltransferase motif. And actually, interestingly, if you look in some gram-positive bacteria, they have genes with the ADA transcription regulatory motif linked to an alkylation glycosylase domain. And there are many more examples of this. Unusual domain patterns in DNA repair genes. If you look at papers by Arabin and Eugene Koonin in particular, they've shown dozens of examples of unusual shared domains between DNA repair genes that no one has studied the function of that particular domain structure. So a thing related to this extra subfamilies of proteins that has come up when doing genome analysis is that we found many examples where there are extra copies of some DNA repair gene in the genome. And they're not identical to each other, so it's not just like a recent duplication of the gene. They're more closely related to the other gene in the genome, or relatively closely related to that other gene, but they're diverged enough to suggest that they might have somewhat slightly different functions. So I'm going to give you a couple of examples of this. One of them came up when we finished the genome of this endosymbiotic/parasitic bacteria called Wolbachia. They do all sorts of interesting things to the hosts that they infect, like cause males to be sterile, or kill male offspring, or do other nasty things, usually to males in the population. And when we sequenced and analyzed the genome, we found the first example so far of a prokaryote, of a bacteria with 2 homologs of mutL. So it has a normal mutS, a mutS1, and this organism has 2 homologs of mutL that are diverged from each other, but more closely related to each other than they are to any other organism that's out there. And it's unclear if this is some sort of initial evolution of some system like in eukaryotes, where there are multiple mutL and mutS homologs, or if this is just these genes do similar functions to each other, and it's not like a eukaryotic system. But there's no experimental data yet on these 2 different mutL homologs in Wolbachia. Another one that I found to be very interesting came up when I was involved in helping analyze the genome of Agrobacterium tumefaciens, which is an organism best known because it causes crown gall tumors in plants, and it's used for genetic engineering of plants. The genome turns out to have 1 large circular chromosome, 1 large linear chromosome, and 2 moderately sized plasmids. That's what makes up the genome of Agrobacterium. And interestingly, it looks like on each of these genetic elements, there's a DNA ligase homolog, possibly a DNA E homolog, and there are at least 2 of them, if not a third, has exo3 homologs. And it's not clear why each of these individual genetic elements would require their own DNA ligase homolog. Is it involved somehow? Does it need to be physically close while it's being transcribed and translated to some site in the genome to work. It's not clear. But this was the first example where there were multiple replicons within a prokaryote, and each of them had a ligase homolog on that replicon. So here's one that I'll come back to later, but I want to introduce here. When the anthrax genome was sequenced at Tiger, we found that there were two relatively closely related but somewhat divergent homologs of the UV dimer endonuclease. I don't believe either of these has been experimentally characterized in anthrax, but it's not clear, again, why you would have two homologs of the UV dimer endonuclease. Again, is there some sort of division of labor into different pathways for repair for these genes compared to the species like S. pombi and Deinococcus and Neurospora that use UV dimer endonuclease for repair? So one of the things that we don't understand how to make predictions about what's going on in the organism all the time is related to what I mentioned for Helicobacter pylori. When you analyze a genome and you don't find homologs of some pathway that you're interested in the genome, what do you conclude? Does the organism not have that process? Or are the genes so divergent that you can't find them? Or has it invented a new pathway to carry out that same process? So I'm going to give you a couple of examples where we don't know what's going on in organisms, but where we've found unusual patterns of absence of particular repair pathways. I've already mentioned the mismatch repair one. That's been found in lots of organisms. I'm going to give you a couple of other examples. So when we were involved, Tiger was one of the sequencing centers involved in sequencing the genome of Plasmodium falciparum, the causative agent of many forms of malaria. When the genome was being annotated, one of the annotators at the Sanger Center wrote me an email and said, there's this unusual DNA ligase in the genome, and he didn't understand what's going on because the automated annotation called it DNA ligase 4, and there was no annotation that annotated a DNA ligase 1. And if you look at experimental studies in eukaryotes, many people would suggest that DNA ligase 1 has more of a core function in ligase activity, and DNA ligase 4, which is primarily involved in non-homologous end joining. So has Plasmodium falciparum done something weird compared to other eukaryotes? So again, if we do an evolutionary tree of the ligase homologs in a variety of organisms, including eukaryotes and viruses and archaea, and group them into subfamilies where this subfamily is the ligase 1 subfamily, here's the ligase 4 subfamily, there's some viral like ligases, and here's an archaeal ligases. If we ask, is the automated annotation of this genome consistent with these trees, it turns out that it's not. So if we look within the ligase 4 subtree, within that tree, we don't see a Plasmodium falciparum homolog. But we do see it in the ligase 1 subfamily. So it looks like, from my point of view, the automated annotation was actually wrong. It made a miscall for what this gene is likely should be annotated as. And this is a good thing because it turns out that there are not homologs of any of the other genes known to be involved in non-homologous end joining in eukaryotes in the Plasmodium genome. So we can't find any homologs anywhere in the genome of these genes. So now the question, which is still not resolved, is does this mean that Plasmodium does not have non-homologous end joining? Or does it use some alternative pathway to do non-homologous end joining? Or does it rely entirely upon homology-based processes to carry out repair of strand breaks? It's not clear, but the absence of the Ku genes, the ligase, all of the genes involved in non-homologous end joining in other eukaryotes is an interesting observation in Plasmodium. So I'm going to tell you another story about Wolbachia and some of its similar organisms in terms of biology. When people have looked at the genomes and genes in these intracellular bacteria, bacteria that live inside the cells of a host, in particular a eukaryotic host, they find that if you look at this tree and you look at the branch lengths of the tree, the branch lengths indicate something about the evolutionary rate of these organisms. And you find that these intracellular organisms tend to have a higher evolutionary rate than free living relatives of theirs. This is true for virtually all intracellular organisms that people look at. These are mutualistic symbionts of insects. We're doing a project on this organism, which is the glassy-winged sharpshooter, and it has endosymbionts that live inside of it that feed it some amino acids that it can't make for itself. And if you look at the endosymbiont tree compared to free-living relatives like E. coli, Haemophilus, Pastorella, you see that all of these organisms have longer branch lengths, meaning that they've evolved at a higher rate than their free-living relatives. Two theories have been proposed to explain why this seems to be the case across the board for most intracellular organisms. The loss of DNA repair genes, or some type of altered population genetics, such as going through a lot of bottlenecks. When a lot of people have looked at this data, not just us, many people have studied this before we even got involved in it, the conclusion has been primarily that it looks like altered population genetics is the explanation, and not loss of DNA repair genes. For example, in the Wolbachia genome, we certainly find a reasonable number of homologs of DNA repair genes, and many of these, this is a similar list to some of the free-living relatives of Wolbachia. So we wouldn't really conclude that there seem to be massive defects in DNA repair in Wolbachia, and therefore we would favor the population genetic explanation for why evolutionary rates have gone up throughout the genome. If we turn to those insect endosymbionts, I know you can't read this, but this is a table of putative DNA repair genes and all those different insect endosymbionts that I showed you before. What we see is that some of them have interesting DNA repair genes missing, but some of them have a full suite of DNA repair genes compared to some free-living relatives. So again, we're not sure if we can prove that they don't have DNA repair defects, but at least because some of the organisms in this group seem to have a lot of DNA repair genes, again, we favor the population genetic explanation. That is, until we look within the symbiont group. So if you look just within this endosymbiont group and we look at which ones have the shortest branch lengths of all the endosymbionts, this is that symbiont of the glassy-winged sharpshooter with a short branch length. These are some other insect endosymbionts with very long branch lengths. It turns out the ones with the longest branch lengths don't have homologs of mut-L and mut-S, and all the other ones do. And the one with the shortest branch lengths, the sharpshooter symbiont, is the one with the full suite of DNA repair genes. So it looks like there is some type of correlation within this group in terms of evolutionary rate and the presence and absence of particular DNA repair genes. And I'm going to come back to this in a little bit, but note, for example, that some of these endosymbionts are missing Rec-A, which prior to genome sequencing projects was thought to be more of a universal type gene across all organisms. So another interesting thing that's come from analyzing these insect endosymbionts and comparing them to organelles. So if you look in Arabidopsis thaliana, the model plant species, and you look for bacterial-derived DNA repair genes encoded in the nucleus, these are likely genes that once were encoded in the organelle genomes and have now been transferred to the nuclear genome of Arabidopsis. We find a variety of homologs of DNA repair genes. Some unusual patterns here, but a variety of homologs. One of them I want to call attention to, which I'll come back later, is that they have homologs of MFD, the transcription repair coupling factor, which, at least when it was originally characterized, was involved in coupling UvrA, B, C, D excision repair to the transcribed strand of actively transcribing genes. So we can't find any homologs of UvrA, B, C, and D in the genome of Arabidopsis. So what is this MFD doing? It looks like it's targeted to the chloroplast. It looks like it's actually derived from the chloroplast genome. But why would you have an MFD and not UVR, A, B, C, and D? And if we go back, I know again you can't see this, but if you look at the insect endosymbionts, many of them have MFD and no UVR, A, B, C, and D also. So These endosymbionts give you a possible model system for studying interesting decouplings of various DNA repair genes that are thought to interact with each other in some model organisms, but maybe this provides an avenue to study what else they might be doing in the absence of some of the genes they normally interact with. So, I'm just trying to get the time. So another thing that's come up related to this MFD versus UVR ABCD pattern is that we find lots of examples like this where DNA repair genes don't always, the distribution pattern does not always match what people thought the pathway was in other organisms. So for example, we find many cases now where there are Rec BCD homologs, but no Rec A in a genome. As I said before, we find MFD, but no UVR A, B, C, and D. There are a variety of other patterns that are sort of inconsistent with either the type of organism that you're looking at or the type of biology. One of them that a variety of people have studied for a few years is that in thermophilic bacteria, and thermophilic archaea in particular, there was no homolog of the normal uracil DNA glycosylase. And this was surprising because when you grow at high temperature, you should have more deamination. If you have a lot of deamination, you probably want to have some pathway for repairing uracil in the genome. So this absence of homologs of the normal uracil DNA glycosylase served as a suggestion to people for pursuing other types of uracil repair in these thermophiles. And again, there are a lot of examples where we find unusual things, such as Rad25 homologs in some bacteria like Mycobacterium tuberculosis. We have no idea, as far as I know, what that gene is doing, but possibly it's this is a pathogen, possibly it's somehow interacting and disrupting the host Rad25 function. I think perhaps the most interesting unusual patterns that are still worth pursuing are what's going on in archaea. So even to this day, there have been very few genetic or biochemical studies of DNA repair processes across the diversity of archaea that are out there. And there are certainly some interesting things that I think people are pursuing right now, but need more research. So for example, many archaea have UVR ABC homologs. Many other ones have homologs of some of the eukaryotic nucleotide excision repair proteins. And then, interestingly, there are some species with both of these. And it's not clear if there are two alternative excision repair pathways going on in these organisms, or if maybe some of the eukaryotic genes are involved in transcription and not excision repair. It's not clear what's going on in these archaea. In terms of mismatch repair, you actually have many archaea that have patterns that resemble what's going on in eukaryotes. So there are many species that have multiple MutL homologs and multiple MutS1 homologs, like with the multiple MSH homologs and PMA-PAL and other MutL homologs in eukaryotes. And again, it's not clear what's going on in these organisms, whether or not they're alternative functions like in eukaryotes. So I've tried to just give you a quick overview of some of the unusual things that keep cropping up when we do genome sequence analysis. But I hate to tell you this, we haven't even started along the path of finding unusual things. And there are two main reasons why I think we haven't started. The first is that the genomes that we have available are incredibly biased subset. They're from mostly very limited types of organisms. Most in bacteria are from 3 phyla, the proteobacteria, the firmicutes, which are the low GC gram-positives, and the high GC gram-positives, also known as actinobacteria. This shows up a little more clearly, the bias, when we look from an evolutionary point of view. This is an evolutionary tree of the 40 proposed phyla of bacteria that were documented a few years ago. Those are the 3 phyla in yellow, where most of the experimental studies where most of the genome sequences are available right now. These new ones that I just showed here, there's 1 or 2 genomes for most of these groups, but very poorly sampled. And you can see there are lots of other phyla in red for which there are not genome sequences even available yet. We have a bunch of projects at Tiger, including an NSF Tree of Life program to start to fill in these gaps. That's what's labeled in blue. So there are many more genomes coming along where there are going to be many more unusual patterns in terms of putative DNA repair genes in the genomes. And I call your attention to one thing on here. There are many that are still shown in red. Those we don't have any genome sequences from because these are organisms, these are phylobacteria for which there is not a single cultured species. No one has ever grown a representative of this entire group. They're only known through ribosomal RNA sequences that have been gotten from PCR and environmental samples. So there's a huge amount of diversity out there that we have not yet sampled. One of the solutions to this is to actually go out and sequence directly from an environmental sample. Some of you may have heard about Craig Venter's trip around the world to sequence the oceans. I've been helping him analyze some of that data where you go and collect a sample of ocean water, filter out the microorganisms and sequence the crud out of them, and then try and analyze everything in that sample. And just to give you a taste of what we're going to find, if you remember I mentioned before that UV dimer endonuclease tree. Here it is again, the UV dimer endonuclease with what looks like some more diversity than had been previously studied experimentally. Here's the new tree of UV dimer endonucleases. Those are the previous ones that I had in that other tree. And these are all the ones that came from one sample in the Sargasso Sea. An unbelievable diversity of UV dimer endonucleases out there in uncultured organisms. This, we get the same pattern for every single DNA repair gene we look at in these environmental samples. Just an unbelievable amount of sequence diversity of homologs of these genes. Again, we don't know if they're repair genes, but they're close relatives of these genes, and we don't know anything about the function of basically any of them. Okay, so now I've told you there are all these questions. How are we going to come up with some answers? I'm just going to provide a couple of quick suggestions of ways to start getting at this. One is to do better bioinformatics, to make better predictions based upon the sequences that are available. And I'm just going to quickly mention one approach to doing this. There's a whole suite of prediction methods that are called non-homology-based functional prediction methods. And they're really interesting. And I'm going to give you an example of one called phylogenetic profiling. Here's how it works. You take all the genes in your organism of interest, you search it against all other complete genomes. And again, you want complete genomes to search against. And you ask, yes or no, is there a homolog of this gene in the other species you're looking at? And then, rather than asking if your gene of interest is similar in sequence to something that someone has experimentally characterized, you group genes by their species that they're found in. That's what this is a cluster diagram of the distribution patterns of genes from E. coli across a large number of other genomes. And red means a gene is present in these other species, and black means it's absent. And you get these nice clusters of genes found in similar species across the tree. Just as a reminder, this grouping genes by distribution patterns can be very useful. For example, MutAs1 is always found in species that also have MutL. MutAs2, that H. pylori gene, has no correlated distribution to MutL homologs. So you can actually predict the functions of genes. In this case, you could have predicted that MutAs2 might not be involved in mismatch repair because it doesn't have a correlated distribution pattern to MutL. I'm assuming you can't see this very well over there, but this is a cluster diagram of a low GC gram-positive, and all of these genes are sporulation genes that are found in other low GC gram-positives, and embedded within this list are genes that no one has ever studied, shown experimentally what it's involved in. So it's found in the same species that all these other Spo genes are found in, and not any other species. So you might guess that that gene is involved in sporulation. Here's another cluster, UVR, A, B, and C. They're always found together, basically, or almost all the time. If you have UVRA, you have UVRB, and you have UVRC across all organisms in the tree. Here's yet another example of a clustering of genes that are found in gamma proteobacteria, including MUC-E, MUC-D, MUC-F, CA, DNA polymerase 3 size subunit, and mute-H, actually, the protein that directs mismatch repair in proteobacteria, but it's not found in most other prokaryotes. So better bioinformatics, I think, will help us make these predictions. And of course, there's one other way to help make these predictions, and I'm going to point this out by telling a story about novelty. So one of the reasons I went to Tiger in the first place is they were sequencing the genome of Deinococcus radiodurans. And, this is, at the time, was the most radiation-resistant organism known. There are some publications of other relatives of Deinococcus that might be even more radiation-resistant. But you can gamma radiate Deinococcus until you get bored, and it doesn't really care. When we were finished sequencing the genome of Tiger, and I went through and looked for homologs of DNA repair genes in the genome, hoping to find that golden nugget of why this organism was so resistant to radiation, and we found this nice list of putative DNA repair genes. It's got everything: mismatch repair, recombination, base excision repair, nucleotide excision repair, ligation, everything you can think of. The problem is, so does E. coli, so does Bacillus subtilis, so do lots of other organisms. They have basically the same list that we found in Deinococcus radiodurans. Why is this? Well, the reason, again, if we go back to that same tree I showed before with the genome sequencing projects, there are at least these 40 phylobacteria. Most of the experimental studies of DNA repair have been done in 2 phyla of bacteria: low GC gram-positives and proteobacteria. Here's Deinococcus radiodurans down here. It's separated from these other lineages by about 2 billion years of evolution. And we're trying to predict its DNA repair genes by looking for homologs of Bacillus and Streptococcus or E. coli genes. It's just not the way to discover novelty in an organism. You don't discover it by looking for things that are shared across all species. So the solution is to start doing experiments in all these other lineages across the tree. And then we'll start to better understand the diversity of DNA repair processes in different organisms and start to better predict the functions of new organisms once we have more of a diversity characterized. And of course, I've focused on prokaryotes. That's where most of the experiments are available and most of the genome projects are available. But the same is true for eukaryotes, the same is true for archaea, the same is true for viruses. We need more experimental studies of organisms either with novel biology or from across the tree. And I will end there. Leave my acknowledgement tree up on the slide there. So that's it.
Where's your new edition?
I took out the picture of my baby because I couldn't copy it from iPhoto.
Okay, thank you. Try the lights on. Okay, let's see how we. Here we go. John, there you are, John. Okay. Okay, you can wait. Yeah, now they can see it. And if you move to the center of it, closer to me. It's coming from a camera over the monitor. Okay. Now let's try. The rule is 1 question per site that will go around. And since we see Baltimore, why don't you unmute in Baltimore and take it away there, Will?
Thanks, John. That was going to strain your voice so much, and I guess it really the sound just made it through. But thanks. It was very interesting. I guess a question right here.
Nice presentation. I wonder if you could rationalize the patterns, gene patterns, based on the lifestyle of the organism, so what they're exposed to in their environment.
We can, for some of the genes, we can rationalize it. So, for example, photolyase homologs, they tend to not occur in deep sea organisms or organisms that don't see a lot of UV radiation, although that's not entirely the case, so we don't know if the photolyase homologs in some of these other organisms are circadian rhythm regulators like they might be in eukaryotes. We can see a variety of genes that seem to correlate with lifestyle for oxygen exposure as well. But the correlations are right now very weak. And one of the problems with doing these correlations is that we've, as a country, as a scientific community, we've dedicated a lot of energy to sequencing all these genomes, but not to recording the physiology and biology of the organisms and putting them in the same databases to do the correlations. So we have about 250 prokaryotic genomes completed and in GenBank right now, but we only have, for example, mutation rate information for 20 of them. And we only have lifestyle in terms of, UV exposure that they might get for another 40 of them. It's very hard to do these correlations without that biology. So we see hints of it, but we don't do the statistics of it yet.
Thanks.
Okay,
thank you. That was Michael Seidman at Aging Institute, and the last that you please identify yourself when you're asking questions next. Let's go to NIEHS, some research triangle.
Jonathan, this is Ben Houghton at NIEHS. Enjoyed your talk very much. Intrigued by the MFD, being in a lot of organisms that don't have UvrA. Do they have prokaryotic RNA telomerase, or do they have just eukaryotic RNA telomerase? Because maybe MFD is working with the transcription machinery.
So the endosymbionts have prokaryotic everything. Prokaryotic RNA polymerase, prokaryotic DNA polymerase. They just don't have UVR, A, B, C, and D. Arabidopsis, is different. It has a mix of the plastid and mitochondrial-derived genes, including this MFD, as well as all the normal eukaryotic genes. So it's possible that that MFD is doing something in the nucleus with the eukaryotic genes, or it's possible it's targeted to the organelles and other eukaryotic genes are targeted to those organelles. So it's much more complicated in Arabidopsis. In the endosymbionts, it's much more straightforward. A lot of them have MFD and no UVR, A, B, C, and D, and then mostly relatively simple transcription complexes.
Okay, thank you. Let's move to SUNY Stony Brook. Anyone there in Stony Brook? How about Brookhaven?
John Dunn at Brookhaven. How can we rule out that some of the hypothetical genes are really the critical components in these systems? Is there a systematic attempt to knock out in a few of these organisms hypothetical genes so we're left with a limited set?
There are systematic efforts in some of the model organisms to knock out the two types of hypothetical genes. So there are the hypothetical genes that have no homology to anything in any database, and then there are the hypothetical genes that have a match to, we call them conserved hypotheticals. They have a match to something in the database, but for which no one has ever studied the function of those homologs. There are systematics in some model organisms, systematic efforts in some model organisms to do that, but in a very limited number of species. And in many cases, as far as I know, there's no one, or very few cases, where people are focusing on mutation and repair aspects of those hypothetical or conserved hypothetical genes. Many of them, they're just looking for lethal phenotypes, for example. So I don't think there's a general systematic effort to do this with a focus on mutation and repair.
Okay, thank you. Anyone there at Livermore? How about University of Pittsburgh?
Yes, we are here. Jonathan, thank you for that talk. This is Rick Wood at Pittsburgh. You made a compelling argument for sequencing other bacterial phyla. On Arabidopsis, they have eukaryotic NER genes such as XBB, XBC, XBF, but there's no XPA in Arabidopsis or any plant as far as I know. Have you been able to detect an XPA or wonder what could be substituting for it?
I confess I haven't looked at Arabidopsis since right before the paper came out in 2000, so I haven't looked with newer methods that might be better able to detect XPA-like genes. I believe there are multiple other eukaryotes that do not have obvious XPA homologs either, so it could be the case where something else can substitute for that function and has in a variety of organisms. So I haven't looked in a while on Arabidopsis. It might be possible to do it, for example, with hidden Markov models, which are a better way for looking for distantly related homologs of genes that we did not use when we analyzed the Arabidopsis genome for the genome paper. So
yes, Gene Conant says he can't find one in plants by that method, so maybe there's just another gene there.
Yeah, so then one of the approaches that we've tried to use in prokaryotes is this phylogenetic profiling. And it turns out right now it doesn't work very well in eukaryotes. One reason for that is we don't have enough genomes, and another is that they may not have the same evolutionary processes that lead to this correlated presence and absence of particular genes. We think a lot of that is due to lateral gene transfer. So in prokaryotes, you can search for missing genes by this clustering genes by distribution patterns, and Kunin and Aravind have actually done that and found some genes that appear to have anti-distribution patterns to a variety of DNA repair genes in prokaryotes. You could do the same thing with eukaryotes, it just may not work very well.
Thank you.
Okay, thank you. Let's go to University of North Carolina, Chapel Hill. There. How about Galveston? Is anyone there? How about University of Kentucky?
Here, this is Dave Warren. You talked a lot about DNA repair enzymes. Can you tell us anything about the distribution of translation filters?
I could try and remember having looked at this a few years ago, but it's not really what I've focused on. I mean, there are a reasonable number of papers out there where people have looked at that. There certainly is more of a diversity of translesion polymerase in terms of the phylogenetic tree of them. Every time we sequence a few more genomes, we seem to find a new subfamily of homologs of those genes. So I think there's an enormous diversity of those out there, but I have not actually looked at the distribution pattern of these across species. We certainly see many cases where there are no homologs of these genes in organisms, and many cases where there are one or two, I think sometimes more, but again, it hasn't been my focus, so I don't have anything more to say.
Okay, thank you. How about, let's go to Oregon, Portland, Oregon.
Yes, Jonathan, this is Stephen Lloyd. I just wanted to ask you, you had commented a couple of times throughout the talk on the UVDE. Have you also done an analysis on the pyrimidine dimer-specific glycosylase AP lyses throughout all the sequencing that's been done?
You mean the ones related to MUC-Y endo 3?
I was specifically referring to those of like the T4 pyrimidine dimer glycosylase.
Yeah. So there are not very many of those that have shown up. In fact, we were involved in a collaboration to sequence another T4 phage genome, which does have homolog of that, and there have been a few found, I believe, mostly in other phage genomes. I, in fact, took out a tree of those at the last minute before the talk. I have not looked extensively for those across all the genomes. We focused on the ones closely related to the T4 one when we did this analysis for the other T4 phage, and there certainly were a reasonable number that had come up in other phage.
Thank you.
Okay, thank you. Let's go to University of Michigan, Ann Arbor. Max, can you get closer to the can you get closer to the microphone, please?
Better. Well, when we've done evolutionary trees of the SNP2 family to try and assign different homologs that we find in genomes to subfamilies within the SNP2 family, we have found examples like Drosophila where there do not appear to be genes that map perfectly into the Caucanes B subfamily within the SNP2 family of proteins. It's not entirely clear if that's due to incompleteness of some of the genomes. So most eukaryotes, they have not sequenced deeply into the centromeres and other hard parts to sequence in the genomes. So it's possible that there's a CSB ortholog there that we didn't find when we looked at the genome. And we've seen this in a few other eukaryotes. It's also possible that the evolutionary trees are imperfect and you get a gene that's actually more closely related to Caucanes B that groups with something else for a variety of statistical reasons. So we haven't really applied the best evolutionary methods and the best alignment methods to confirm or deny that, which is why we haven't published a new paper on the SNP2 family and all the subfamily distributions across species. It looks like it's missing from many species, but we're not certain.
Okay, thank you. Is anyone there? Wake Forest? Okay, did I skip anyone? It's a burning question. Okay, does anyone have any questions? Yes, Dimitri. Go ahead, Dimitri.
Anybody else try to use this information about the variability for interacting proteins, MAT-S, MAT-L, or some proteins from nucleotides in repair to identify interacting interface, look at co-evolution. You showed an enormous amount of sequencing data, and each pair has been developed in evolution and has been actually selected from millions of years. So it's maybe really unique material to understand protein-protein interaction.
I couldn't agree more. We are doing that exact thing with some of these genes. Actually, I think a large number of people are doing the same thing. In particular, people working on protein-protein interactions, like you mentioned. So there are two ways that you can look at this. You can look at the co-evolution of individual amino acid residues in the proteins, and you can also look at co-evolution of just bigger picture things like domains or a variety of other superstructures in the proteins. And there are a large number of people that are just starting to do this. The genome data turns out to not be quite enough to do this for most genes. We've tried it and the statistics are not really there. However, this environmental sequence data might start to provide us with enough to look at co-evolution of amino acid residues. So for example, for REC-A protein, which I've done some of these co-evolution studies of the filament and monomer contact regions of the crystal structure, there are now about 5,000 homologs of REC-A from this environmental data, as opposed to about 5 complete REC-As in GenBank for it right now. So I think when you add all of that extra data, you will be able to do it. But right now with the genomes, we may not have enough signal to do it just with that data.
Okay, Dimitri? Okay. Questions here? We want to thank you very much for coming by.